notice something about this. This integuments are covered on all sides. Can you see? It will be covered like this. Entire thing is covered except this top part over here. This top part is left free over here. Can you see? This is called as the micropyle. Okay. Integuments encircle the nucellus except at the micropyle. So this top part over here, the micropyle, will be left free open over here. The integuments do not fuse over here into each other. They leave it open. This is the micropyle. Okay. And this side of the ovule is the micropylar pole. Okay. Later, when we study the division that happens within the ovule, you'll have the micropylar pole, you'll have the chalazal pole. Okay. So remember that the side, the micropyle side, is known as the micropylar pole. All right. Now I told you that the integuments protect the nucellus. Okay. Why does it need to protect the nucellus? What is the nucellus actually? So nucellus is a mass of cells, okay, and it has a lot of food reserve. Okay, it has abundant food reserve. Okay, because if you see inside the nucellus, you have the next structure, which is the embryo sac. Can you see the structure here? Yes. So inside the nucellus, you have the embryo sac. Okay. Embryo sac is where all the division will happen. Okay. And this embryo is finally going to grow bigger. Okay. And this is how you're going to get the new flower or the new plant. Correct. So to give nutrition to this embryo sac, the nucellus is present around it. Okay. It has food reserves. All right. Which is why this has to be protected. Okay. That's why you have two integuments, inner and the outer integument. Now, located in the nucellus, like I just told you, is the embryo sac, which is also known as the female gametocyte. Okay. The embryo sac is also known as the female gametocyte. Okay. Now, opposite to the micropyle end, this is the micropyle end, right? This opening that I told you. Opposite that, you have another pole, which is known as the chalazal pole, which is nothing but the basal part of the ovule. Okay, so this is the base of the ovule. All this structure is inside the white thing that I showed you, in the previous diagram. This one, right? This is there, right? So all this, what I'm telling you, is within this, okay, because it is nothing but the structure of the ovule. Okay, so all this is within that white colored structure that you see. Okay, so are we clear? We have the funicle, we have hilum, we have integuments. There are two integuments, remember this. Okay, which are the two integuments? You have the inner integument and you have the outer integument. All right. Then you have the micropyle. Okay. The micropylar pole as well. Then you have the nucellus, which has the food reserve. Okay. Finally, you have the embryo sac. So embryo sac is the innermost part of the ovule. Correct? And to protect the embryo sac, you have the nucellus. I mean, to give food to the embryo sac, you have the nucellus. To protect the nucellus, you have the integuments. Correct? Finally, you have the funicle, which through which the ovule is attached to the placenta. All right, is this clear? This is the structure of the megasporangium or the ovule. Now you have another concept here called as the megasporogenesis. You remember in anther, we had in the pollen grains, we had something called as the microsporogenesis, correct? So in this case, in megaspore, you have the megasporogenesis. So here, the formation of the megaspores occurs through the megaspore mother cells. Okay. So here it is called as MMC. Correct? In this case, it is the megaspore mother cells, right? What is it in case of microsporogenesis?
Pollen mother cells.
Yeah, pollen mother cells. So there it is abbreviated as PMC. Here it is MMC. When you write your answers and give your exams, don't mix up the two, all right? So generally, a single MMC is differentiated in the micropylar region, okay, where the megaspore mother cell will undergo meiosis or meiotic division. Okay, so where is the micropylar region in this? Here, this top end over here, right? This is the micropylar pole. So around this region, okay, is where you're going to see the megaspore mother cell undergo meiosis. Okay, so what will meiosis result in? It results in the formation of 4 megaspores. Okay, so here you get 4 megaspores from 1 mmC. Okay, 1 mmC will give you 4 megaspores. Okay. Now, out of these 4 megaspores, 1 megaspore is functional, which means it works, it will carry out all functions, it will be all right. Whereas the other 3 are degenerate, okay, these are not functional, okay, and these will just disperse off. Only one is useful, okay, or functional out of the 4 that are formed. Now, only the functional megaspore will develop into embryo sac or the female gametocyte. Okay. So, out of the 4 megaspores, only one is functional, and this one functional megaspore will then develop into the embryo sac. Okay. So, in this diagram, the innermost part here is the embryo sac, correct? This is the embryo sac. So from where will this embryo sac be formed? This will be formed by one functional megaspore. Is this clear? Okay. Embryo sac, yeah, only the functional megaspore will develop into embryo sac or the female gametocyte. Okay, so this form of development from single megaspore is known as monosporic development. So since the embryo sac is formed from just one megaspore or a single megaspore, we call it as a monosporic development. Here again, mono means one, correct? And it is formed from one megaspore, right? So that is why it is called monosporic development. Is it visible?
Yes.
Okay, I'm going to play this. You can read as the text comes on the screen, and you can also observe the animation. So basically, now they're going to show you the structure of the ovule, okay, and how the female gametocyte is, um, how it develops. Okay, so I'm going to stop the video till wherever we've done today, all right, because it has some other extra information as well, which we will see in the next class. You all remember this stage? The megaspore mother cell will undergo meiosis to give you 4 megaspores, right? So each of this is haploid, each of this is an N. Remember this? The 3 megaspores will degenerate and only one will remain functional. Okay, so what has happened here? The one functional megaspore which remains, it will grow in size to become the embryo sac, okay, which is the innermost layer. Okay, so